Hi, I'm Christian and I'm here to talk about Pfeiffer syndrome. Pfeiffer syndrome is a genetic disease that results in various developmental abnormalities mainly related to improper skull suture closure. This can result in eye bulging, midface hypoplasia, bread thumbs, soft tissue syndactyly of hands and feet, and great toes. Development under these conditions can result in the following complications: reduced brain development, which results in mental disability; obstructive sleep apnea, which can result in suffocation while sleeping; corneal ulcers, which can result in blindness; and seizures. The disease was first characterized by Rudolf Pfeiffer in 1964. Pfeiffer syndrome is caused by mutations to the fibroblast growth factor receptors 1 and 2. 25 mutations account for 60 to 70 of the instances. Three types of the syndrome denote various degrees of severity and stem from the functional consequences of the mutations to the receptors. Type 1 is associated with mutations in both FGFR1 and FGFR2. FGFR1 mutations only account for a small fraction of the type 1 population. This is the mildest form of Pfeiffer syndrome. Type 2 and type 3 are associated with mutations solely in the FGFR2. Type 2 is the most severe, displaying a pronounced skull abnormality. Type 3 has intermediate severity with respect to the other subtypes. Pfeiffer syndrome displays an autosomal dominant inheritance pattern. This means that one mutant copy is enough to cause the disease in a person. This suggests that two functional copies of each FGFR receptor is required to maintain normal signaling. FGFR1 is located on chromosome 8 and FGFR2 is located on chromosome 10. These mutations in these genes occur sporadically since most severe cases result in poor treatment outcomes. Figure 2 on the bottom right is a series of known mutations that cause Pfeiffer syndrome that are the result of single amino acid changes. The main treatment is corrective surgery that intends to provide some relief to the intracranial pressure. It is performed at various stages in the patient's life as development continues. Prognosis is highly dependent on symptom severity. There is currently no treatment that can regain function of the null FGFR. With an increased understanding of the FGF pathway and the structural biology behind the FGFR stability and functionality, a druggable target may one day be developed successfully. Alternatively, if recombinant DNA technology increases its precision to a standard fit for human use, FGFR mutations could be screened and rescued in the zygote stage. Nonsense mutations could be repaired using Cas9 nicases coupled to base end editing domains like adenosine and cytosine deamidases. The FGF receptor family has 4 members. We are most concerned with the receptors 1 and 2. These receptors, though, all bind to 23 different FGF ligands, all with different affinities and specificities towards particular receptor families. FGF receptor 1 has a greater affinity for FGF1 subfamily and FGF4 subfamily. FGF receptor 2 has an affinity for FGF7 subfamily. If we take a closer look at these 3 subfamilies, we can see that various things happen when they are mutated in organisms. The interesting thing about it is that FGF3 and FGF2, which are primarily bound by the receptors 1 and 2, both play a role in skeletal development. The FGFR family belong to a group of proteins called the receptor tyrosine kinases. 2 FGF receptors dimerize in the presence of a ligand and phosphorylate each other at a number of sites. Any 2 FGF receptors can dimerize. The ligands determine which pairs join, so it's most likely that ligands are tissue specific. This results in a signaling cascade that activates the STAT and ETS transcription factors while inhibiting FOXO1. Now I'd like to talk about a paper that investigates The impact of FGFR mutations throughout skeletal development in mice. Development is a complex system that involves the integration of multiple signaling pathways. Researchers wanted to see the impact that FGF mutations had on bone development, and to do this, they had to identify changes to other signaling pathways in the system. Two of the major other signaling pathways are Wnt signaling and BMP signaling. They're mainly known to cause ventral dorsal patterning recognition in the developing organism, but also play a role in later stage development. An important protein to keep in mind is axin 2, which binds to beta-catenin and marks it for destruction. BPN signaling results in the expression of a transcription factor called SOX9, which plays a strong role in skeletal development and inhibits endochondral ossification at later stages. To develop the mouse mutants, the researchers used the Cre-Lox recombination system. This system works with two components, the LOX sites and the Cre recombinase protein. The LOX sites are placed in between the genes that you wish to target. The Cre recombinase gene is placed in a promoter specific to the tissue you wish to target. When these components are combined in a developing organism, it results in the targeted deletion of your gene of choice only in the tissue you choose. Various staining techniques were used in these mice for microscopy. H&E staining were used for histology, and hemotoxin stains nuclei blue, eosin stains extracellular and cytoplasm pink. Alcium blue was used to label chondrocytes. Alkaline phosphatase staining was used to label osteoblasts. Beta-galactosidase staining was used to label knock-ins, since they used the beta-galactosidase gene to knock in a gene in order to take away its function. Immunofluorescence was also used to label various proteins, and tunnel assay was used to identify the degree of apoptosis going on in the developmental stages. Once the mouse lines were developed, they were allowed to grow for a variable amount of days in order to capture all the critical stages of their growth and development. After this, the skull tissue was excised and various staining techniques were used to aid in the microscopy. A collection of mesenchymal stem cells were used to run additional experiments in vitro and then run by microscopy again. The images and data generated from the microscopy was analyzed and then interpreted to generate conclusions. The upregulation of Wnt signaling brought upon by the disruption of axin 2 and the downregulation of FGF signaling brought upon by the disruption of FGF1 resulted in a greater generation of chondrocytes. And this is shown in the three figures I presented. The first one on the left showing 100% of the double mutants having excessive chondrogenesis, which you can see with the white regions in the center of the images. The figure on the left shows that the double mutants had higher expression of SOX9 and type 2 collagen. And the AB staining on the bottom left showed an increase in chondrocyte proliferation in the double mutants. These mutations to the Wnt and FGF signaling pathway was enough to result in craniosynostosis, which likely is occurring through ectopic endochondral ossification, which is the calcification of cartilage into bone. This can be shown in the azar and red stained skulls, which is a staining chemical that targets the ossified extracellular matrix that often surrounds bone tissue. And it shows that the double mutants clearly had a much more pronounced stain right in the sagittal suture. In the figure at the bottom, we can see the histology of the sagittal suture, where it shows populations of the ectocranial and endocranial tissue in the double mutants, which is similar to the wild type posterior frontal sutures, but not the sagittal sutures, showing a change in morphology. The figures to the right, you can see that the double mutants progressively lost chondrocytes in A to C while increasing osteoblastogenesis in J to L. In the middle dark figures with the tunnel assay and the laminin, you can both see increase in apoptosis in the tunnel assay and increase laminin in the immunofluorescent image. Maruyama et al. Then developed a conditional knockout of FGFR1 driven by an axin 2 Cre promoter so that FGFR1 would only be disrupted in the presence of axin 2 expression. This was confirmed two different ways. First, by using an FGFR1 primary antibody to determine if it was successfully knocked out. And two, by identifying the presence of beta-galactosidase, which was the insert that was knocked into the ORF of the fibroblast growth factor receptor. The specificity of disruption can be seen in the top figure to the right that shows the mouse skull, where the control has no markings, but the knockout has clear blue borders around all the plates of the skull. Using this system, AB staining was used to reveal an increase in chondrocytes in the PF of the FGFR1 knock-ins, but no change in the sagittal sutures. Using the mesenchymal stem cell cultures collected from the mice early on, AB staining was used to identify the balance between beta-catenin and fibroblast growth receptor signaling's effect on mesenchymal cells. BMP signaling was found to increase in the PF suture for double mutant, with SOX9 expression dramatically increasing. In the figure on the bottom right, it shows that noggin significantly suppresses chondrogenesis, while BMP increases it. This makes sense since noggin is a strong inhibitor of BPM. In conclusion, beta-catenin is required to maintain an undifferentiated state. The presence of AX2 allows for the normal differentiation of mesenchymal stem cells to osteoblasts and chondrocytes. FGF signaling promotes endochondral ossification, but its absence has no effect on skeletal precursor expansion. FGFR1 deficiency enables BMP signaling to stimulate chondrocyte maturation. This increase in chondrocyte is believed to promote premature bone tissue formation, but no mechanism was suggested. This research highlights a hint of the mechanism going on in Pfeiffer syndrome, but has many limitations. The main one is that craniosynostosis instances mainly involve mutations in FGFR2. The study of FGFR1 is inconclusive in the context of skeletal development if most of the Pfeiffer cases involve the receptor 2. Also, the complete aberration of FGFR1 is not a great model of Pfeiffer syndrome biology. With most cases involving missense mutations, the targeted mutagenesis of FGFR1 may have resulted in a different characterization. Thank you for watching.